Okay, so now we want to talk about meiosis. Um, and let's begin. All right, meiosis. Meiosis is a type of cell division that is going to be responsible for reducing the number of chromosome sets per cell. So if we are reducing the number of chromosome sets per cell, we know that this is already different from mitosis, because mitosis was making an identical copy. Okay, so meiosis, we're reducing the number of chromosome sets per cell. So my question to you is, why would we want to reduce the number of chromosome sets per cell? Absolutely, because when sperm meets egg, sperm needs to have half. Ovum or secondary oocyte needs to have half. So that when their nuclei fuse, the parent has the offspring has the same identical number of chromosomes as the parent had. So if we didn't reduce the number of chromosome sets per cell, then that new organism that's being created is going to have way too much DNA, and it's going to have some problems. So this process is also called reduction division. We know that normal cells have a diploid set of chromosomes, so they're normally 2N. If we use us in an example, we have 46 chromosomes, so that's 2 times 23 will give us 46 chromosomes. 23 of those chromosomes coming from mom, 23 of those chromosomes coming from dad. So we know that the sex cells have to have half of that original DNA material that are in normal cells. So when producing sperm cells, when producing a secondary oocyte, or what we call an egg, but it's officially not an egg until it's been fertilized, but what we call an egg, it has to have half of the genetic material, so that when, again, the two meet together, that's when you form a diploid organism. Okay, so they join to form 46 chromosomes. That is 2N. 2 times 23. 23 paternal chromosomes and 23 maternal chromosomes. All right, so one of the things that you'll notice, or if you've read the chapter, looked in the book or anything, you'll see that when producing these cells, none of them are identical. So in other words, let's just look at in your family. Even if your sister or is not an identical twin, then you guys are going to look somewhat different, because every single time your parents make a sperm or a secondary oocyte or egg, that there's some variety because of the processes that take place during meiosis that don't take place in mitosis, that makes those cells different. So it says people are unlikely to produce 2 identical gametes, 2 identical sex cells, 2 identical egg, or 2 identical sperm cells for the men in their lifetime. So if we were to consider human males as an example and look at their ejaculations, per ejaculation, we have 300 to 500 million sperm cells in that one ejaculation. So that's about 120 million sperm cells per ml. Okay. Now, if we multiply that by a lifetime of ejaculations, then we would have trillions of sperm produced, yet it is highly unlikely that 2 of them that have been produced over that lifetime are identical. So the question is, why? What is going on in meiosis that makes those cells that are being produced different? What's happening in meiosis that didn't happen in mitosis that's producing these different daughter cells? So we know that each of us is unique and genetically distinct unless we are identical twins. And identical twins, we know, are formed from the union of one egg, one sperm, that within the 2 weeks prior to the differentiation process, when cells become specialized, that embryo splits into two separate dividing masses of cells, which means that they're going to have what? Identical of whatever mom donated, identical of whatever dad donated, because we started with the fusion of this sperm's nuclei and the fusion of the nuclei of the secondary oocyte. And when they fuse together after the first week, but prior to the second week's completion, it says that those cells have the same DNA, but they split into two separate dividing masses of cells. So again, unique and genetically distinct. So let's look at the meiosis phases. We're going to have meiosis number 1, and in that we'd have prophase 1, metaphase 1, anaphase 1, telophase 1, followed by a brief phase in between called interkinesis, and then we'll have meiosis number 2, which will have prophase 2, metaphase 2, anaphase 2, and telophase 2. So let's begin with prophase number 1. In prophase number 1, it says the chromatin net coils to form who? Chromosomes. We've seen these things happen before. The nuclear membrane, it's going to do what? Disappear. It's going to dissolve. The nucleolus, it's going to disappear or dissolve. The spindle fibers are going to form as those centrioles are going to divide and move to the opposite poles of the cell. So here are those centrioles that have moved to the opposite poles of the cell. Now, big thing that's happening. We said, where is the variety coming from? Well, we see two things on here. It says synapsis occurs in prophase number 1, and crossing over occurs immediately following synapsis. So, what is synapsis? Synapsis is the pairing and bonding of homologous chromosomes, which you can see, which is happening here. These guys are paired up and they're bonded. The blue one we say is paternal. The red one we say is maternal chromosome. Okay? Then it says crossing over is going to occur. So again, synapsis, the pairing and bonding of homologous chromosomes. So what are homologous chromosomes? Homologous chromosomes, they have the same size. They have the same shape. They're also going to have the same genes. Okay? So we're going to find, so what you just saw in that last slide was that the matching maternal chromosome and the matching paternal, I should change colors if I'm able to find the pen. It says that they find each other and they bond with each other. So the matching maternal number 1 finds the matching paternal number 1 all the way down through all of the chromosomes. So the 2 number 1s, 2 number 2s. Okay, for example. So again, we said they have the same size, same shape, same genes, and they also have the same centromere location or position. It says, so we end up with what's called a tetrad. So here's a tetrad. So here's one chromosome we can see. Here's the next chromosome. Then we got one of the blue ones, so that's 3. And then the last one here, that's 4. That's a tetrad, 4 chromosomes or 4 chromatids, okay? Then it says that crossing over occurs. Crossing over is the exchange of chromatid segments. And when they exchange these chromatid segments, they change in terms of their alleles. Okay? So, again, we will draw, here's the paternal chromosome I have to find. And here's my maternal chromosome. Well, it says when they pull apart, they've exchanged some chromatid segments where the paternal chromosome is no longer all paternal, nor is the maternal all maternal, but they have bits and pieces. So I'm going to draw one of these over here. So if this is the maternal chromosome and it's pulled away, then this chromosome, find my pen, will share, well, it pulled apart part of dad's chromatid and genetic information. Same thing with dad. Where he pulled away, he will also share and have some of mom's DNA. So now these chromosomes that were completely paternal and maternal are now partly maternal and paternal in terms of their alleles. So you can see that now we have these new combinations of alleles produced following crossing over. So this is a shuffling process that produces most of the variety that you're going to see in meiosis, because think about it, when these guys pull apart, they're all going to move into different gametes. So, for example, this one will be in a gamete, this will be in a gamete, this, and so that means that every last one of these is going to be different. Okay, every cell that's produced is going to be different. Now, I can't do it the way that it's happening in the body, but my little cartoon drawing is the best that I can do. All right, so this also shows you that same thing. And if you notice here, they've exchanged, where they were all crossed over, they've exchanged some information in more than one place, unlike what I produced for you in my cartoon drawing. All right, so what is a gene versus allele? So it says alleles are then alternate forms of the same gene. So people may look different even though they have the exact same parents, and it's because you don't know what genetic information is being passed along. So even though, say for instance, if we're coding for hair on the head, well, there could be a gene that says you're going to be bald, okay, and there's a gene that says you're going to have lots of hair. But depending on which gene you got is going to depend on, or which set of genes you get is going to depend on what you actually look like. So we can use this as another little play example of hair color. Say there's a red allele, a black allele, or blonde allele. It says all of these are coding for what? The color of hair, but they're all different, okay? So different forms of the same gene. All right, since I've run out of time, I will stop here, and I'll cover the rest, some more phases in the next video. Have a good one.